Well, hello everyone. I hope you all are well and taking very good care of yourself. I welcome all of you to the very first biology lecture of the KVPY series. As you know, it is a very prestigious examination and biology is an integral part of it. To all of the KVPY aspirants, I would recommend to go through all my videos because elaborate detailed study of the biology is the key, right? So keep watching those videos. Well, as part of this KVPY series, what we are going to do in these slots or in these classes is we are going to discuss the previous year questions, and I would emphasize on explanation so that with every question you get to learn some of the theory of it. For the detailed part of it, you should go and keep on watching the videos. Well, so as a part of it, today I'm going to start with the previous year question paper, that is KVPY question paper of 2019, precisely the SA 2019. And in this lecture, I will be covering the first part of the biology. Okay, so without much ado, let's start. Okay, great. Well, I would also like to add here before starting is biology part is easier, as in if you know, you can quickly finish it up, so it will be saving your time. At the same time, it is very scoring. So take benefit of it, okay? Keep on studying and you can score a lot so that it can improve your overall rank, okay? So from the 2019 paper, essay code, this is the first part of the biology. Now, this part contains 15 questions, that is from 46 to 60. So let us deal one by one. The very first question, okay? Let me first. Okay, so now everything is fixed. Let us start. So the very first question, that is question number 46, says that which one of the following is a correct statement about primate evolution? Okay, now the very first question is primate. Okay, now we are primates. Primates are a group of mammals, okay, which have opposable thumb, okay, which have opposable thumb. Like in primates, there is human beings, apes, orangutans, gorilla, gibbons, monkeys, these all are primates, okay? So the question says that you have to pick the correct statement regarding the primate evolution, as in how the primate evolved, okay? Let us see the options. The first option says chimpanzees and gorilla evolved from macaques, right? Now, macaque is a type of monkey. Macaque is a type of monkey. So this says that chimpanzees and gorillas have evolved from macaque monkeys. Okay. What is the second option? Second option says humans, chimpanzees evolved from gorilla. So it says that humans and chimpanzees have evolved from gorilla, as in gorilla was our ancestor of humans and the chimpanzees. Okay. Well, the third option says that human, chimpanzee, and gorilla, all these three evolved from a common ancestor. Okay, great. And the last option says that humans and gorillas evolved from chimpanzees. Well, if you know the evolution chart of primates, then it's good. If not, let us look at it, and there we can find the answer. Okay. So what is here on your screen is the diagram showing the evolution history of primates. Okay. Now, can you see? Here are the humans. Okay. Here is the human. This is bonobo monkey. This is chimpanzee, gorilla, orangutan, and gibbons. Well, remember, your C option said that humans, chimpanzees, and gorilla evolved from a common ancestor, right? Can you see here this ancestor called hominine Okay, this is called hominine, is the common ancestor for humans, bonobo monkey, chimpanzees, as well as gorilla. Okay, great. So if you know this chart, you know the answer. The correct answer would be C, and all rest of the options are wrong. Correct? So see, chimpanzees and gorilla have not evolved from macaques, rather they have commonly evolved from hominine. Similarly, human and chimpanzee have not evolved from gorillas, right? So the correct answer is C. Okay, so option third, that is C, would be correct for it. Now, in order to answer such questions, you need to learn these evolution diagrams. Okay, these are called cladograms. So these cladograms I will give you in the video lectures, or even I have showed right now. You can make this cladogram and learn it. If you do not remember it, you can't answer such questions. So it is very straightforward. If you know it, you do it in no time. Like it will take hardly 10-15 seconds to do such a question. Okay, great. So moving on to the next question. So this is the Hindi part of it, the same previous question. Okay, so question number 47. Now this question says, the crypt of Libercoon, right? The crypts of Libercoon are found in which one of the following parts of the human digestive tract? Right? So the question is asking about a particular gland called crypts of Libercoon, and the question asks, where is it found in the human digestive tract or human digestive system? Well, is it found in esophagus or oesophagus? Both are the same thing. Or in small intestine, or stomach, or rectum? Correct? Now, to answer this question, you need to know the histology of the human digestive tract. Okay, this is part of human physiology, especially the digestive system. Now let me tell you, the correct answer is small intestine. Okay, now in the small intestine there are these villi like structures. Okay, and within these villi, there in this area there are some glands. These are called crypts of Libercoon. Okay, now this Libercoon is named after the scientist who discovered it. So in intestine there are two types of glands found: crypts of Libercoon and Brunner's gland. Okay, now since the whole agenda of these videos are not to tell you the correct answer, rather to give some insight, some information regarding these topics. Okay, so let me tell you that in small intestine there are these two type of glands, crypts of Libercoon and Brunner glands. Out of that, Brunner's gland is only found, or I should say glands, these are only found in duodenum part of the small intestine. Okay, however, the crypts of Libercoon is found all across the small intestine. Okay, so crypts of Libercoon are found throughout the small intestine, while Brunner's glands are only found in duodenum. And these glands are responsible for the secretion of intestinal juice, and this intestinal juice is also known as succus entericus. Okay, आप लोग इस चीज को याद रखेंगे, succus entericus, and this succus entericus, that is intestinal juice, right? Intestinal juice or succus entericus, it contains a lot of enzymes. Okay, it contains a lot of enzymes, such as it contains intestinal amylases for the digestion of carbohydrate. It contains intestinal proteases for the digestion of oligopeptides, okay, or dipeptides, which are broken down part of proteins. Then it contains intestinal lipases for the digestion of lipids or fats, and it also contains intestinal nucleases for the final digestion of nucleic acids. Okay, so this intestinal juice or succus entericus is secreted mainly by the crypts of Lieberkühn. Okay, and these are found in the pits of the villi. So this is villi, and these are the pits of villi. Okay, so it is present in only the small intestine part of the human digestive tract. It is not present in esophagus, not in stomach, not in rectum. Now, since we are talking of esophagus, let me tell you, esophagus contains no glands, none. Okay, small intestine, you know it now. Stomach, it contains gastric glands. Okay, it contains gastric glands, while rectum, again, it has some glands, but we are not talking here. So for you, there are no glands. Okay, great. Remember? So the answer would be small intestine. Great. Let's move to the next question. Oh, okay. Okay. Well, so next is question number 48. This question says, removal of the pancreas impairs the breakdown of, right? So let me simplify this. The question says, if pancreas is removed from an organism, or I should say, if pancreas is removed from a human, it will lead to the impairment of breakdown of which of the following. That is, अगर हमने pancreas को किसी human being से निकाल दिया तो उसकी body में किस किस चीज का digestion ठीक से नहीं होगा. ओके, impair का मतलब problem, ठीक है, ठीक से नहीं होना. Breakdown is for digestion, क्योंकि आप जानते हो digestion is the process by which complex food material is broken down into simpler molecules, right? So basically it is a task of breaking down. तो क्वेश्चन है कि अगर हमने पैनक्रियास को रिमूव कर दिया तो किस किस चीज का ब्रेकडाउन ठीक से नहीं होगा, यानी कि किस किस चीज का ठीक से डाइजेशन नहीं होगा. वेल, टू आंसर दिस यू शुड नो कि पैनक्रियास ऐसा क्या सिक्रीट करता है, और जो कुछ भी सिक्रीट करता है वो किस किस चीज के डाइजेशन के लिए इंपॉर्टेंट है या किस किस चीज का डाइजेशन करता है, राइट? वेल, इफ वी आर टॉकिंग ऑफ पैनक्रियास, okay, let me use this space. Well, if we are talking of pancreas, pancreas secretes a lot of enzymes. That is, it secretes pancreatic amylases. Now, the amylases are required for the breakdown of carbohydrates. Okay, plus it secretes pancreatic peptide, pancreatic proteases. Okay, proteases such as trypsinogen, such as chymotrypsinogen, such as procarboxypeptidase. These all are type of pancreatic proteases. Now, since these are proteases, they are responsible for the breakdown of proteins, that is digestion of proteins. Not only this, pancreas also secrete pancreatic lipases. So these are responsible for the digestion of lipids or fats. Okay, great. Apart from this, pancreas also secrete pancreatic nucleases, which are required for the digestion of nucleic acid, that is DNA or RNA. Okay, DNA and RNA. Great. So this pancreas actually secrete a whole lot of enzymes, all types of enzymes required for the digestion of carbohydrates, proteins, fats, and nucleic acids. Correct? So if pancreas is removed from a person, it will impair the breakdown of all of these following: carbohydrate, protein, fat, as well as the nucleic acid. Now let's go back to the question and try to solve it. Lipid and carbohydrate only? No. It will also impair the digestion of proteins and nucleic acid. So this is wrong. Lipids and proteins only? Obviously this is also wrong. Lipids, proteins and carbohydrate? Yes. There is no only, so this statement is right. Proteins and carbohydrates only? This is also wrong. Are you getting it? So the key word here is only. So when you are solving such questions, do pay attention on such small words. They look as if they are insignificant, they makes no sense, but actually they makes a lot of sense. They will make you fall as a prey, make you fall as a victim. ठीक है, तो इस ट्रैप में आपको नहीं पढ़ना है और जो ऑप्शंस है उनको ठीक से पढ़ना है. ओनली मेक्स अ लॉट ऑफ डिफरेंस. ठीक है, सो करेक्ट आंसर वुड बी सी. Okay, so answer number 3. Okay, let's move to the question number 48. Sorry, 48 we have done. Move to the 49. Well, what is this question? Let's see. It says microscopic examination of a blood smear reveals an abnormal increase in the number of granular cells with multiple nuclear lobes. Which one of the following cell types has increased in number? Okay, see, first of all, it is a piece of advice for all of you for any competitive exam, or for that matter for any exam. The first thing you need to do is to keep your head cool and calm. Do not buckle up. Okay? Cool and calm. With this cool and calm head, you can think straight and you can minimize your errors. Okay? So first thing, keep your head straight, keep it cool and calm, and read the question carefully. While reading the question, do not keep your clock ticking in your head. Do not. Because when the time bomb is ticking in your head, most probably the ability to comprehend the question is not right. It does not work well. Okay, so just for that time being, when you are reading a question, stop your time bomb. Do not let it deviate. Okay, focus on the question, read it carefully, very carefully, and then most of the time you will make correct options. If you don't know it, if you have not read it, that is altogether different scenario. But if you have, then and you are reading it with a straight head, with cool, and with all of your composure, most probably you will make the correct you will choose the correct option. Okay, great. So we are going to read it. It's a long sentence. So microscopic examination. So obviously what you are going to do is you are examining through microscope, and what you are examining is a blood smear, right? Now many of you may ask, what is a blood smear? आप लोगों ने कभी blood test कराया है? If any of you have gone through a blood test, and if you have seen the pathologist doing it, you know that they take a glass slide, something like this. Okay, बस वो glass का बना होता है, तो something of the shape and size, there is a glass slide, and on this glass slide they take a drop of your blood, put it here. Now still that blood is in the form of a drop, right? In order to watch it properly under the microscope, they want to make a thin film of the blood so that all the cells are arranged in a single layer, so they can watch it carefully, they can watch it properly. Okay, so what they do is, with the help of another cover slip or maybe another slide, they make a monolayer. They make a, they take this drop and make it a flat, spread it on this glass slide. Okay, so basically they spread it to make a thin film of the blood, and this is called blood smear. Okay, so basically spreading it is called blood smear. So the question is saying that suppose you are a student or maybe a pathologist and you are examining a blood smear, and when you are watching it, what you found in the examination is there were abnormally high number, means very high number of some granular cells with multiple nuclear lobes. Okay, आंसर यहीं पे है. अगर आपको मालूम है तो आंसर आपको मिल चुका है, राइट? सो बेसिकली आपको बहुत ज्यादा नंबर्स दिख रहे हैं किसी पर्टिकुलर टाइप ऑफ सेल्स के, और ये जो सेल्स हैं वो ग्रैनुलर हैं विद मल्टीपल न्यूक्लियर लोब्स. नाउ द क्वेश्चन सेज व्हिच वन ऑफ द फॉलोइंग सेल टाइप हैज इंक्रीज्ड. सो बेसिकली दे आर आस्किंग which cell has granular cytoplasm with multiple nuclear lobes. That's it, simple. Okay, they are asking the morphology. Well, the option says lymphocyte, monocyte, neutrophil, and thrombocytes. Well, first of all, these all are blood cells because it is a blood sample, right? And secondly, these are granular cells, and you know that only the WBC cells have granular cytoplasm. So obviously this will be the any one type of WBC. Well, if we see the WBC, see here, WBC, that is white blood cell, we find that WBC are mainly of two types: granulocytes and agranulocytes. Okay, now granulocytes because ये साइट का मतलब होता है सेल, और ग्रैन्यूल का मतलब होता है छोटे 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 ऐसे पार्टिकल्स, insoluble particles in the cytoplasm, right? So there are a lot of granules in the cytoplasm. Okay, great. And because of granules, these cells get stained very easily. They get stained very easily, that is they catch up the color and they become colored. They can be either yellow or red or blue. Okay, so depending on the staining dye. Okay, so these are granulocytes because their cytoplasm has these granules, plus they have this nucleus. Okay, and in the nucleus obviously they will be having this genetic material. Correct. Now these granulocytes are further of three types: acidophil, that is also known as eosinophil, basophil, and neutrophil. Okay, we will see later. And the agranulocytes are further of two types, that is monocytes and lymphocytes. Okay, now we will talk about each of them in detail when we will be learning about the animal physiology. Okay, in animal physiology we will be learning about this blood profile. There I will be discussing in great detail about all these things, but as of now what I'm going to do is I'm going to show you the diagram of each of these cells. Okay, great. Well, what you see here are the three different type of granulocytes, right? Eosinophil or acidophil, basophil, and the neutrophil. Now what you notice here is the cytoplasm of all these cells have these granules, right? And hence they are granulocytes. Secondly, if you focus on the nucleus, you see the nucleus is not the usual spherical nucleus, rather it is lobe-like, that it is lobular. You see the eosinophil or acidophil, it is bilobed. The basophil, it is also kind of bilobed to trilobed. But if you see the neutrophil, it is four-lobed. Usually the cells of neutrophil have 3 to 5 lobed nucleus. Since there are more than 3 lobes or 3 lobes or more, we say it is multilobular nucleus. Okay, great. Great. So now you have seen the structure of all these different type of granulocytes, right? So the question says the number has increased of a cell which has granular cytoplasm with multiple nuclear loops. So what it would be? Neutrophil, right? So answer will be neutrophil. Answer is third, C. Great. Moving on to question number 50. Well, 50. Let's see the question. The question says, which one of the following genetic phenomena is represented by the blood group AB? Okay. Now, for this, you need to have the knowledge of genetics. I will be covering genetics in good detail because it is very, very important topic, as well as very interesting topic. Interesting and very important because a lot of questions are asked from this topic. Okay. However, this topic is taught in class 12. So if you want to read it, you have to watch the videos of class 12 whenever I'll be teaching genetics. Now, in order to make you understand, I would require you to recall some of the basic concepts of genetics that you all must have learned in class 10th. Okay. Now in class 10th, you have studied about the Mendel experiment, and there you know that there were 3 laws of inheritance given by Mendel. That is law of dominance, law of segregation, and law of independent assortment, right? Great. The first law, that is law of dominance, says that one allele is dominant on the another, right? So when both alleles are present in an organism, one allele will express itself, the other will not be able to. Correct? So the allele which expresses is dominant, one which is not able to express itself is recessive. Well, this is the concept of Mendel, but this concept is not always true. In many cases, it fails. Okay, the example is the inheritance of the blood group in humans. Okay, in humans, the inheritance of blood group does not follow the law of dominance. Okay, now how it happens, let us see. Suppose a person has 2 alleles for its blood group, that is A and B. Both alleles they have. As per law of dominance, if A and B both are present, then any one should express itself only. So the person should either be A or B. But what we find in human population, the person is AB. That is, both the alleles express themselves, right? Now, this type of inheritance, this genetic phenomena is known as co-dominance. Co का मतलब क्या होता है? साथ में, जैसे कि co-ed होता है, co-education, जहाँ पे कि मेल एंड फीमेल, सो बॉय एंड गर्ल्स दोनों साथ में पढ़ते हैं, दैट इज कोएड स्कूल्स, बिकॉज को का मतलब होता है साथ में, राइट? एंड डोमिनेंस मीन्स बोथ आर गोइंग टू शो देयर प्रेजेंस, करेक्ट? नाउ so it is the case of codominance, right? Now there are other phenomena as well, like incomplete dominance. So in incomplete dominance, one allele is only able to dominate incompletely. There is no complete dominance. Okay, we will see all these things. Incomplete dominance we see in case of antirrhinium, where red and white flower blends in to give a third character, that is pink flower. Okay, so this all we will see again in the genetics. As of now, you just remember that the AB blood group or ABO type of blood group that is found in humans is an example of codominance. Okay, at the same time, in humans, there are not 2 but 3 alleles that govern the blood type. Okay, usually so far in class 10th, when you have studied the genetics, Mendelian genetics, you only have seen 2 alleles for each character, such as capital T and small t, capital R small r, capital Y small y, and so on. But there are certain characters for which more than 2 alleles are found, and such a character is the blood group of humans. So there are 3 alleles that governs the blood group in humans, and those are small I, capital IA, and capital IB. So these are the 3 alleles which govern the blood group. So if a person has both small I, they will be having O type of blood group. If they have IA and small I, or IA, in both cases they will be having A blood type. If they have IB small I and IB, they will be having B blood type. And if they have both IA and IB, then there will be case of codominance and blood group will be AB, right? So the blood group inheritance in humans is an example of codominance. At the same time, since there are 3 alleles, it is an example of multiple allelism, okay, or multiple alleles. ठीक है, याद रखिएगा इस बात को. In detail, I will describe the whole process, how it happens in the genetics when I'll be teaching this particular topic in standard 12. Well, so moving on to the next question, that is 51. Let us see the 51 question. This question says the mode of speciation mediated by geographical isolation is referred to as. Well, first of all, this question belongs to evolution. Okay, so this question is coming from the evolution chapter. In order to fully understand this question, you should know what speciation is. Okay, now speciation is the process of creation of new species, new type of living organisms from the existing organisms. Okay, that is called speciation, right? जैसे कि हमने first question में देखा था कि from a common ancestor called hominine, the humans and the bonobo monkey and chimpanzees, okay, these all have originated. So from one species there are 4 species coming or originating. Now this process is called speciation, that is creation of new species from the existing species. Okay, and this in the process of speciation, the role of geographical isolation or reproductive isolation is crucial. Okay, now again, this we will be talking in the evolution section, but as of now, the speciation is of different types. Okay, mainly allopatric, parapatric, and sympatric. These all are type of speciation on the basis of geographical isolation. Okay, well, if the speciation occurs because of geographical isolation, that is, 2 populations are isolated by some geographical barrier, either a sea or a big river or mountain or things like that, then we call it allopatric speciation. Okay, if the speciation does not occur because of geographical isolation, मतलब ये 2 जो species बन रहे हैं, they are evolving in the same geographical area, तो हम उसको बोलते हैं sympatric speciation. Okay, now you will ask what is parapatric. Now parapatric is because of the formation of smaller population at the periphery of a larger geographical area. ठीक है, तो ये सब हम लोग वहां पे डिटेल में देखेंगे. बस याद रखिएगा कि speciation which occurs due to geographical isolation of the 2 population, we call it allopatric, है ना. If If the speciation occurs within the same geographical area, we call it sympatric. Okay, so for this question the answer should be allopatric. Okay, great. So B. Well, question number 52. This question says, which one of the following metabolic conversions require oxygen? ठीक है, अगेन ये क्वेश्चन आप सबके लिए इजी है, आप लोगों ने 10th में पढ़ रखा होगा. यू मस्ट हैव स्टडीड अबाउट रेस्पिरेशन, है ना, आपने रेस्पिरेशन पढ़ा होगा. एंड यू नो दैट रेस्पिरेशन इज ऑफ टू टाइप्स, दैट इज एरोबिक एंड एनारोबिक. एरोबिक एंड एनारोबिक, ठीक है. और एरोबिक में ऑब्वियसली O2 इज रिक्वायर्ड. Here O2 is not required. You know it. Aerobic and anaerobic. Now, in aerobic respiration, you know that the glucose is completely oxidized in presence of oxygen into CO2, H2O, and ATP, that is energy. Right? Whereas the anaerobic respiration is of two types. One is called ethanolic fermentation and second is called lactic fermentation. Now in ethanolic fermentation, the glucose is oxidized in the absence of oxygen to produce ethanol plus CO2. In lactic, glucose is oxidized in the lack of oxygen to produce lactic acid only. Okay, here also 2 ATP is produced, here also 2 ATP is produced per ATP molecule. Right? Now if you have this knowledge of respiration, now we can answer this question. So question is, which one of the following metabolic conversions require oxygen? कौन सा? Aerobic respiration. And aerobic respiration में क्या बनता है? Glucose to CO2 and H2O. Right? Glucose to pyruvate is glycolysis. This is called glycolysis. ये सब type of means this occurs in all type of respiration and this is an anaerobic process. इसमें oxygen का use नहीं होता, so this is wrong. Glucose to CO2 and ethanol, this is ethanolic fermentation. Again इसमें भी oxygen का requirement नहीं होता है. Glucose to lactate, इसमें भी नहीं होता है because this is lactic fermentation. Correct? So correct answer will be B. Now moving to question number 53. Well, now 53. This question says, where are the proximal and distal convoluted tubules located within the human body? Right? Now it is a question you are able to answer if you recall the structure of nephron, the structure of kidney. Well, let me draw for you here. In kidney, okay, let me make kidney for you first. See, this is how a kidney looks like. Bean shape, if you recall from class 10th. It is a bean shaped organ with a ureter coming out from here, which goes and attaches to the urinary bladder. Now, if you see this kidney, the kidney is divided into 3 areas. Okay, this is called cortex, this is called medulla, and this is called pelvis. Okay, so kidney is divided into 3 parts: outermost cortex, middle medulla, and innermost is pelvis. Now the nephron looks something like this. Okay, these diagrams are not very good, but you can see in this in the book. So this is one nephron. Well, this part is called PCT, proximal convoluted tubule, and this is called DCT, distal convoluted tubule. So the question is asking, where are the proximal and distal convoluted tubules located? right? So PCT and DCT. Now, this entire part is found in the cortex. This part, which is loop of Henle, this is loop of Henle, is found in the medulla. Okay, so now you can answer this question. The proximal and distal convoluted tubules are found in the renal cortex, into renal cortex, because this cortex, the part of kidney, are called renal. Okay, so this is renal cortex, this is renal medulla, and this is renal pelvis. Okay, so the answer would be renal cortex, C. Now, some students, if even they know and they are not paying proper attention, they are in a hurry, if the time bomb is ticking in their head, they might go blind for this adrenal and they may tick adrenal cortex. Okay, just don't fall prey of it. ठीक है, आपको ये नहीं करना है, ठंडे दिमाग से सोचना है. एड्रिनल कॉर्टेक्स एड्रिनल ग्लैंड का पार्ट है. यहाँ पे हम किसकी बात कर रहे हैं? किडनी का. ठीक है, जो नेफ्रॉन है वो किडनी का फंक्शनल यूनिट है, तो ये कहाँ होगा? किडनी के अंदर, इन रीनल कॉर्टेक्स, नॉट एड्रिनल कॉर्टेक्स. ओके, एड्रिनल ग्लैंड इज प्रेजेंट अबव द किडनी, और उसमें भी होता है एड्रिनल कॉर्टेक्स एंड एड्रिनल मेडुला, पर वहाँ पे अलग चीजें होती हैं. एड्रिनल कॉर्टेक्स में जो fasciculata, reticulata एंड ग्लोमेरुलोसा होता है. ठीक है, वहाँ पे नेफ्रॉन्स नहीं होते हैं, वहाँ पे पीसीटी डीसीटी की कोई बात नहीं आती. ओके, सो आप लोगों को बायो में ध्यान रखना होगा, ऑप्शंस केयरफुली पढ़ने होंगे. ग्रेट, सो रीनल कॉर्टेक्स. मूविंग टू द नेक्स्ट क्वेश्चन, क्वेश्चन नंबर 54. वेल, okay, now this question is again a very good question. You have to apply a lot of brain, but again this question is from standard 12th, genetics, right? The question says that in a diploid organism, when the locus X is inactivated, transcription of the locus Y is triggered. Based on this observation, which one of the following statement is correct? It is a very good question, and it requires a deeper understanding of the genetics. Well, let me try to make you understand. The question says that in a diploid organism, so the organism is diploid as in it has 2 copies of each chromosome, right? So each chromosome is present in 2 sets. Hence it is diploid, that is 2n. Great. Next it says when the locus X is inactivated, transcription of locus Y is triggered. Based on this observation, which one of the following statement is correct? Well, so here we are obviously dealing with the 2 genes. One gene is X, the other gene is Y. Now, this X gene may have 2 alleles, that is capital X and small x. At the same time, this capital gene also may have 2 alleles, that is capital Y and small y. So we are going to deal with the 4 alleles, and we have to see how the phenotype is affected when these 2 genes are present in different combination. Well, when there are 2 genes and their phenotype, the outcome is dependent on both the genes, that is both the genes are interacting with each other. This interaction could be positive or can be negative or can be neutral. Okay, but this 2 is not neutral, either positive or negative. Okay, so these 2 genes are interacting with each other. When the genes are interacting, then we call such a phenomena as epistasis. Okay, many a times we find that the function of one gene is controlled by the other. Okay, as in, let me first tell you one example. In case of pea plants, the flower color purple, okay, especially in sweet pea, Lethyrus odoratus, we find that the purple color flower, okay, let me show you. So let me first delete it. Okay, so we see that the purple color flower is because of a pigment called anthocyanin. So anthocyanin is a pigment which gives the purple color to the flowers in case of sweet pea. Now this anthocyanin we are writing here as capital P for anthocyanin, purple color, is produced from a precursor, say for X. Okay, now this X is formed from another precursor, say A. Now from A to this pro molecule is catalyzed by a gene called C, and then X is formed into this anthocyanin purple color by another enzyme called P. So P is controlled by a gene P and C enzyme is controlled by a gene called C. Now, if there is a capital C, then only functional enzyme will be formed and X will be formed. Again, if there is only P, then only this purple will be formed and the flower color will be purple. If any of this gene is a small cc or small pp, right, in both cases the color of the flower would be white because this anthocyanin will not be formed. Is it clear? Because if capital C is there, then only X is formed. If there is a small c, this will not be formed, so will not be the purple color. If there is this X is formed because of capital C, but here there is only small p, no functional enzyme, again the X will not be converted into anthocyanin, so no purple color. So in either case, if both the alleles for both the genes are not, if both the genes are not having at least one dominant allele, then purple color will not be formed. Correct. Now such a gene interaction where the phenotype is as a result of the function of two genes, we call such a phenomena as epistasis. EP ka matlab hota hai above, stasis means to stand. So in this, one gene is standing over the other in terms of its expression, isn't it? Now if you look at this question, again we will discuss it later. It's a very good question, but we will be able to understand only when I have already taught you the other concepts of genetics about different type of gene interactions. Okay, so for the time being, you just take the answer. Okay, slowly in class 12, after finishing this first unit, I will start teaching the genetics, and there you can watch the video and learn about this whole concept of gene interactions and epistasis. Okay, so for the time being, I'm asking you to be patient. Well, so if you see here, these two genes are again interacting, X and Y, and it is like, if you have understood the C and P concept, it is something like the Y gene is here, which is making one product. Sorry, let me make it here. Okay, so Y gene is here, which is making one product, A to B, and then B is the substrate for the X, which is making the final product, right? So here the X is epistatic to Y is epistatic to X. Okay, so therefore the answer would be D. Well, rest details we will see later. Well, moving on to question number 55. Which one of the following sequence represents the correct taxonomical hierarchy? Well, this I'm not going to tell the answer. Rather, I'm going to tell you the taxonomical hierarchy. It is kingdom to phylum or division, okay, then to class, then to order, then to family, then to genus, and finally this is genus, species. Okay, so this is the order. Now you can do this question on your own. Well, moving to question number 56. Yeah. Which one of the following organ is not a site for the production of WBC? Kidney. Bone marrow? Yes, WBC is formed there. Liver? Yes, it is formed during embryonic condition. Spleen? Again, it is formed. So, it is the kidney where WBCs are not formed. So, answer would be B. Great. Moving to next question. Question number 57. Which one of the following anatomical structure is involved in guttation? Okay. Now, in order to answer this, you need to know what guttation is. Okay. Guttation. I hope you all know transpiration, that is loss of water in the form of water vapor. Okay, so in plant, water is lost in the form of water vapor from the stomata of the leaves, from the lenticles of the stem, or other parts of the plant. Now, this loss of water in the form of water vapor is called transpiration. However, in some herbaceous plants, small herbaceous plants, what we find that there are these water droplets hanging from the leaf margins, है ना, पत्तों के किनारे से ऐसे लीफ से water droplets hang हो रहे होते हैं in the early morning hours. Okay, now this is actually the loss of water in the form of water droplet, right? एक water drop गिर रहा है, so the loss of water in the form of water droplet is called guttation. This is called guttation. Well, now guttation क्यों होता है? देखिए, when root absorb water from the soil, they absorb this water, and because of the osmotic pressure, there is a pressure applied by the root that pushes the water and minerals up into the xylem. Now this pressure, which is pushing the water into the xylem from root up to the stem, is called root pressure. Well, so you think it as if the root has a small pump. Okay, now this pump is pushing water upward, right? Now, if the rate of transpiration is higher or equal to the rate of this root pressure, then there will be no guttation. There will be only transpiration. But think of the condition where root pressure is higher than the rate of transpiration. What will happen? The water in the form of water droplet will start to come out from the different pores of the plant, and this phenomena is called guttation. Now, why it happens? When it happens, again, this we will see in more detail in the plant physiology chapter. But as of now, let me tell you that transpiration is a type of evaporatory process, right? And evaporation depends on the temperature, on the humidity, right? Now, in the early morning hours when temperature is low, when humidity is high, but root pressure is also high, at that time, the rate of transpiration is lower as compared to the root pressure, and hence in small herbaceous plants, water starts to drop from specialized pores in the leaf called hydathodes. This is called hydathodes. Okay, so the answer of this question is hydathodes. Okay, great. Moving to the next question, that is 58. Well, the question is, which one of the following parts of the eye is affected in cataract? Well, cataract आप सब लोग जानते हो, बहुत सुन रखा होगा. Cataract में क्या होता है? Let me tell you. जो आंखों के अंदर लेंस है, उस लेंस में क्लाउडिंग होने लगती है बिकॉज ऑफ सर्टेन चेंजेस। उसका जो मटेरियल है लेंस का, उसका रिफ्रैक्टिव इंडेक्स चेंज होने लगता है, ठीक है। अब सिंस रिफ्रैक्टिव इंडेक्स चेंज होगा तो लाइट में नॉट पास थ्रू प्रॉपरली, और इसकी वजह से जिसको कैटरैक्ट होता है उसको चीजें साफ नहीं दिखाई देती हैं, धुंधली दिखाई देती हैं। एंड इफ द कैटरैक्ट कीप्स ऑन ग्रोइंग, जब बहुत अधिक हो जाता है, द पर्सन मे नॉट सी एट ऑल, कंप्लीट ब्लाइंडनेस में आकर, ठीक है। सो ये एक्चुअली डिजीज किसका है? लेंस का, लेंस ऑफ द आई, correct? So I hope you get the answer. Which one of the following part of the eye is affected? Lens. Well, moving to question number 59. Well, which one of the following organism is a bryophyte? Well, देखिए, this is a question from the classification. Well, the plant kingdom is divided into Thallophyta, Bryophyta, Pteridophyta, Gymnosperm and Angiosperms. Okay. Now, Bryophyta के examples आपको याद रखने पड़ेंगे. Bryophyta का example है liverwort. It is actually a type of Hepaticopsida या liverwort. Bryophyta को फिर से हम further divide करते हैं into liverwort, hornwort and true mosses में. Okay. So liverwort is a type of bryophyta, ठीक है। वॉलबॉक्स is a algae, thallophyta में आता है, green algae है ये। Chlamydomonas again green algae है, unicellular है ये, monerans है। Fern is pteridophyta का member, okay। So the answer is liverwort. इसको हम Hepaticopsida में रखते हैं। चलिए, next question is 60th, that is the last question of first part of this paper, biology section. During oogenesis in mammals, the second meiotic division occurs. So question is asking the oogenesis, that is formation of ovum or egg, ठीक है। So to be precisely egg. So the formation of egg in mammals, this question is particularly asking about humans. So in humans, the oogenesis, that is formation of this egg cell, occurs, okay, and it occurs in the ovary, but precisely it is asking the second meiotic division. Well, if you look at the oogenesis process, which I am already teaching in class 12, so you can go and see the videos. So far I have taught the male reproductive system. Soon I'll be teaching the female reproductive system, and as part of that I will be teaching the process of oogenesis. Now in oogenesis, when ovum is released from the ovary, it has still not completed the second meiotic division, that is egg cell has not formed, okay. So it is formed only after the fertilization. Why? Because in order to complete the second meiotic division, it requires centriole, but the ovum does not have centriole. So when this goes into the fallopian tube and it fertilizes with the sperm cytoplasm contains one extra centriole, and this centriole then helps in the completion of second meiotic division of the ovum, and finally the egg cell is formed, and then the syngamy occurs, and further rest of the processes follow. So the oogenesis in mammal, during oogenesis in mammals, the second meiotic division occurs after fertilization, and it happens because it requires centriole, which is provided by the sperm. Okay, great. So where this will happen? What will be the site? It will happen in the fallopian tube. To be precise, it will happen at the junction of ampulla and isthmus of the fallopian tube. Okay, so please, I would really recommend all of these KVPY aspirants to keep watching my 11th and 12th videos, because there I'll be teaching these all processes in good detail. Here we are just discussing the questions. So watching those videos will help you, okay, will give you the better perspective. So this was the last question. With this, we have finished the first part. In the coming video lecture, we will be covering the second part of this particular paper, that is 2019 KVPY SA paper, that is intended for class 11 students. So till then, keep studying, keep good, take very good care of yourself. We will meet in the next video lecture. Till then, goodbye.